Hi, everyone. I'm honored to be the last speaker of this. Okay, yep. So hello, everyone. I'm honored to be the last speaker at this conference. I know you all are very tired, but it's just 15 more minutes of structural mass spectrometry talk. So today I will be presenting you a part of my PhD project. So I'm working on structural protein analysis and drug design with mass spectrometry. So this talk is going to focus not just at one ligand and one protein at a time, but working on a larger scale. So identifying protein ligand interactions in a complex environment. So I don't think I need a big introduction for the importance of protein ligand structure in drug discovery for this particular group. As you know, 70% of FDA-approved drugs target proteins, and then there is a vast potential in the proteome that is ligandable but hasn't found its drug yet. So according to the Human Protein Atlas, 4,500 proteins are druggable, but only 15% of them have their perfect match yet. So there is both a vast potential of finding ligands for these drugs and potential novel therapies, but also this underlines the importance of protein structure and understanding it for identifying these novel therapies. So there are several classical approaches in the field of structural biology. So these three elephants, X-ray crystallography, which is a classical golden standard of structural biology with high resolution, then NMR spectroscopy, given insights into protein dynamics, and given a medium level resolution. And then cryo-EM, a new and continuously emerging method that you've listened today on, that gives also quite a good resolution and gives also some insights on protein dynamics. But all of these three methods, they have their own limitations. So for instance, for X-ray, one needs proteins to form crystals with a ligand, and for cryo-EM, the instrumentation is quite costly. So ideally, one would need to combine several complementary methods to study the protein ligand structure. And also all of the aforementioned methods, they require purified protein samples. So this is a major hurdle for designing ligands for them, but also it doesn't give a broader picture into how the ligand would interact with other proteins in human proteome apart from its potential target. And then there is a whole other way to look at protein ligand interactions. So this is chemical proteomics, something that my research group is specialized in. So here we're looking at proteome-wide interactions that a ligand can make, which has several pluses, such as no need for purification of the proteins, and one can capture both direct and indirect effects of binding and capture potential off-targets of the ligand. But unfortunately, with classical methods of chemical proteomics, one does not get information on the location of the binding, so where does it happen? And then here, my PhD work comes as a bridge between these two worlds of classical structural biology and chemical proteomics. So what I'm trying to do in my work is to develop methods that would still work with a large proteome, so in a complex environment, but retain some properties of structural biology. So they would be able to give you an insight into where the ligand is binding on the protein structure. So the field of studying protein structure of mass spectrometry is continuously evolving, and there are several ways how one could look at it with mass spec, starting with HGXMS and partial proteolysis-based methods, going to chemical cross-linking, where one is binding together several parts of the protein using these chemical reagents as distant constraints, or using native mass spectrometry to also study protein complexes and stoichiometry. So today we're going to focus a bit more on the method in the field of partial proteolysis. So the method that I have been working on during my PhD at Karolinska and ALLODD is named above filter digestion proteomics. So how does it work? So here on this scheme, I have presented the target protein in two conditions. The one where there is no drug, and so control sample, and then the treated one. And in red, I have colored the peptide in the binding site. So when this peptide is involved in binding with a small molecule, it's harder for a protease to access it and cleave it. So, and this sterical hindrance, or maybe some stabilization caused by ligand binding, is a phenomenon that we use in partial digestion-based proteomics. So what happens then? So when we apply trypsin to digest this protein, and it's in a complex environment, so together with all other proteins extracted from human proteome, we see that in the control sample, this peptide will be cleaved faster than if we compare it to the case where it's treated, so with a ligand around. And then with using low molecular weight cutoff filters and then taking out from the solution all the peptides that have been cleaved during one particular hour, and then repeating this procedure 8 times. So following the digestion of each peptide from human proteome during the 8 hours, we can map their relative peptide abundance, so of each peptide in control and treated samples. And then by detecting the difference in this relative peptide abundance by the center of gravity for control and for treated, we can find which peptides were affected by binding and then link them to potential protein target and also the structure, so the location of this peptide on protein structure. So we used several drug target systems to test this method. So firstly, it was with methotrexate, a classical drug used for chemical proteomics as a proof of principle. Then we chose two brother or sister drugs, rapamycin and FK506. So the reason of choosing them was because they both target the same family of proteins, but then they induce different downstream effects. So for rapamycin, it targets mTOR as an end target through complex formation with FKBPs, and FK506 then inhibits the colurine molecular pathway. So we wanted to see if using the two similar drugs that bind to the same binding site in this FK506 binding protein family, if we could see some differences in mode of action of both of them. And then lastly, we used staurosporine to compare this method to some other chemical proteomics methods. So here I'm showing some of the results of this study. So these are the first 3 drugs that we tested the method on. On the left, you can see the methotrexate data. And as you can see there on these volcano plots, a lot of peptides coming from DHFR, a target of methotrexate, came out as the ones affected by methotrexate binding. So we could identify DHFR as a target of methotrexate. And then for rapamycin and FK506 data, we saw in both cases a lot of FK506 binding protein family members for both of them. But then there were some differences. For instance, mTOR, the end target of rapamycin, showed up correctly only for the rapamycin experiment. And then FKBP3, which showed a lot of targeted peptides in the rapamycin experiment, has much higher affinity to rapamycin than FK503. So we could capture this difference with ALLODD. And then, So as promised, this method should also have some level of structural resolution. So for this, I tested it by trying to use 3 of the top peptides from targets detected by ALLODD by their p-value, and then calculating the center of mass from these 3 top peptides, and then comparing it by the center of mass of the actual binding site found from the ligands from the crystal structure. So here I'm showing some of these cases, and for all of the cases studied, the difference between these 2 center of masses was within 10 angstrom. So while this is not ideal in terms of structural biology, but it can still give an indication for the domain where the binding is happening for larger protein ligand complexes. And also we have used MD simulations to validate some of the findings of ALLODD. So let's go back a couple of slides. So here, for example, for the FK506, so we expect that the peptide affected by binding in the binding site, it will show longer, it will be longer for the protease to cleave it. So it will show on the right of this volcano plot. But then for FK506, we saw 2 peptides coming from a target that showed the opposite trend. So and they were also here, you can see them on the protein structure, but they were quite far from the original binding site. So then together with one of the ALLODD partners, UCB, during my secondment, using MD simulations, we tried to investigate the effects that might be happening in APO and HOLO structures. And we found that upon ligand binding, those areas that showed up in ALLODD become more flexible. So this can indicate some sort of allosteric effect induced by binding. So as a summary, we have identified known targets with high statistical significance using this method, and it could distinguish between drugs with overlapping targets but different downstream interaction pathways. It could be useful in the future for detecting allosteric structural changes, but needs more data to build upon. And it can also give an indication of the binding domain. So with this, I would like to thank you, my group members, my supervisor, and the members of the ALLODD network for supporting me on my PhD journey. And looking forward to your questions that you might have.
I was wondering if you were trying other proteases except for trypsin, if they would give you a better result resolution-wise.
So this was tested in the beginning of the project, trying to use, so first of all, for this kind of work, one needs to use only the specific proteases. This is kind of the limitation for the further use in mass spectrometry, because then you need to know where the protease is cleaving your protein for doing database search. And yes, we tried other proteases, but trypsin gave the best results in terms of sequence coverage and digestion efficiency, so we just proceeded with that one.
Thank you for the presentation. I wonder because you spec would have effects associated with PTMs that you would see in your data.
So for this method, we're not specifically looking for post-translational modifications.
Thank you.
Thank you very much for my talk. I want to ask you when you said you calculate estimate what binding site needs to do this by calculating the center of mass of three peptides. So how do you select these three peptides? Because I guess, first of all, you have to have three that you have found. But then if you find more, I guess depending on which one you choose, the result might be slightly different.
So to distinguish the target, we're using kind of two dimensions or two parameters to do that. So it's the shift. Let's go even further back. So you can see this figures. So first we're using this shift between control and treated samples as one dimension for analysis. And then we calculate the p-value for each peptide between control and treated samples to identify the statistical significance of the analysis. So then we have these two dimensions. And for selecting the peptides for binding site identification, we're using just the p-value as it was producing the best result for this sake. So the top three peptides from the same protein that showed up, that passed the thresholds for the p-value and COG. Yeah, so this is how we calculate.
Thank you for very so. I understand that you work with setups, and the question would be how if you can also study like complexes, more just looking at a protein ligand complex, maybe you can have like monomers versus dimers, so even more complex situations where we have different proteins aggregating.
Yeah, definitely. Just so for this sort of analysis, we need so here we're looking at the difference, right, between the control and treated. So if some protein complexes or complex formation would be affected by the ligand binding, we would see that. Yeah. Was that the question? Or?
He also not strictly related to ligand, right?
Yeah.
Changes the formation of multi.
Yeah, exactly. And we also, this is for the future, but we also think this could be useful for studying protein-protein interactions. But then again, you need this differential condition. So then you need to kind of pre-select which protein you want to study. So the interactions, for example, of cereblon and its protein-protein interactions. So then to have the condition with and without extra protein added to cell lysate.